welcome to the course on biological data analysis and visualization with R. In the last class, we introduced linear models, and we have also seen hands-on how to apply simple linear models to biological data. In this class, what we will be focusing on something called predictive models. Again, we will see how we can build predictive models with linear regression and how this could be useful, right. So, this is something that will be covered in this class. We will be talking about predictive models with linear regression, and we will be also doing the hands-on analysis. So, what do we mean by predictive linear models? So, what we want to do when you build these models, linear models, we want to predict the value of the dependent variable from the values of the independent variables. Okay. So, what it means, we take a data set where we have these xy values and we build this model, right, where we estimate these coefficients w's and the bias term b, and this is something what we call the training data. Okay. So, we build the linear model with a set of values which we call as training data, and then we predict the response, right, and we take another set of values, test data set, right, where we again have these xy pairs, and we take those x values and try to predict y values and see how well do these predictions match with the actual y values. So, this is what we do with prediction with a new set of values. We take the new x values and try to predict the y values and see how well the predictions match with the actual values. So, this is what we call the test data. Now, how does this predictive models help, right, in biological data analysis? So, this is something that will give us insight, right, how well the independent variables predict the dependent variable or the response variable, and this also can give us insights or help us identify the independent variables that are very important for the prediction. Okay. So, this is something in biology, when you think about this, we kind of learn about these factors that are likely to be key drivers of a biological process or a specific function. Okay. So, this is where this predictive linear model will help, and this is something we will illustrate when you go into the hands-on, right, and we will try to predict the expression noise based on all the independent variables that we had in the data set, right. So, we have seen this data set in the last class. We will be working on that same data set, and we will try to build predictive linear models in that process. So, as I said, in the hands-on, what we will be doing is we will first separate out the training data set from the full data set that we have. We will get the test data set out as well, right. So, what we will do in this hands-on is, instead of getting two different data sets, which if we can get, that would be great, but we have one data set. So, what we will do is we will just take out training data set and separate out test data set. We will first build the model, the linear model with the training data set without using the test data set. After we have built the model, then we will take the test data set, we will see the prediction, right, how good the outcome is. Okay. And this is something that will help us identify some of the key features of those models, right. This is something will relate to the biology of the system. Okay. So, let us now move on to the hands-on part, and let us go into the modeling and see how we implement this. Okay. So, here we are in the R console. I just wanted to show you the data that we had already loaded, right. Just to remind you, this is the dimension, and let us just see some of the columns, first 10 columns that we have in the data. And if you remember, right, we had this DMYPD and DMSD. These are the variables that we want to include, that we want to predict, right. These are the noise, expression noise values that we would like to predict from the other independent variables that we have, right. And we have a lot of independent variables. As you can see here, we have about 245 independent variables and 2 dependent variables, right. So, we will predict only one dependent variable, and the similar thing we can do with the other dependent variable. Okay. So, now let us go into the data analysis part. The first part would of course be to generate this training data set and the test data set, right. Separate out this data that we have into training data and test data. Okay. and let us do that. So, what I am going to do is, okay, so what I will do is I will first choose only okay. So, I will choose this, let us say I will keep only one noise value, right, only one dependent variable, and for that so okay. So, that is why I reran this input data tap filtering, so that we end up with only one dependent variable DMYPD. Okay, let us not go into the two dependent variable model. Yeah. Okay, so that let us check the dimension again. It will change a little bit, I think, just 246. So, we have one dependent variable and 245 independent variables. Okay, so let us do this analysis on this input data now, and the first thing is to separate out this training data and the test data. The first command that will run for doing this is identify the training index data. So, we identify these indices which will be used for generating the training data or separating out the training data. And what we will do in this process is we will take out 80% of the full data and consider that as training data, and the rest 20% would be our test data. Okay, so you can think about these different breakups, right? You can have 80-20, or you can have 90-10, or 75-25, or 70-30. But usually this is 80-20 is a good number. So we'll stick with that. But you can of course vary this depending on the data set that you have. If you have a really huge data set, you can of course go for 90-10, so on. Okay, so let's define this index. So what we are doing is we are taking this sample, and out of this number of rows that we have, we are picking sampling, right, randomly picking 80% of them. Okay, so that will define the indices for the training data. What we have in this training index is the indices, the data sets that have been sampled, right, the genes that have been sampled, those row numbers. Okay, so we have 2160 genes, and these are the row numbers that will go into the training data. We are looking into the first six of these samples. So now once we have defined this training index, or we have sampled this, so this is something we can run, right, multiple times, and these values will vary, right, because this is a sampling process. So this will vary from one sample to another. So the next thing is the training data, okay, where we are doing this, taking this input data and considering only those rows that are part of this training index. Okay. So 80% of the data will come here. We can check the dimension. Okay, as you can see, 1728 genes are now here in this training data. The features are of course there, the dependent variable and the independent variables. Once we have identified the training data, we can identify the test data very simply by saying input data minus training index, right, whatever is there in the training index except those, take all the genes in here. And the dimension of this test data, as you can imagine, it will be something around 432. So the total is 2160, right. So that is the total data set that we have. So we have sampled out the training data and the test data. Okay, the dimensions will always remain the same, but if you do the sampling, this training index sample, this command, this will actually sample different genes in different runs. So if you run this iteratively, you will get different sets of genes in the training data and the test data. Okay, so that is fine. Now what we will do is we will build the linear model. Okay, and this is multiple linear regression. We have one dependent variable and multiple independent variables. So let us run this regression model, but the data that we will use, so we are using all features. This is the dependent variable DMYPD. We are running on all independent variables, right, colon dot, and the data is training data. Okay. This is very important. This is where we are going into the predictive modeling part. We are not building this linear model on the full data. We are building this model on training data. Okay. And let us see the summary of this, and what you get is something like this. Okay. So here are the estimates, etc., right, estimate values, which means the coefficients, etc., intercept and coefficients for each of these features. and then also have these p values, right, which features are significant, right. Now they are in multiple lines, you can see, and then you also have these stars, etc. So which means significance of each of these features. What we will be looking at is this adjusted R square. So multiple R square is 0.8859, but the adjusted R square, which penalizes for number of features that are used here, is 0.56. Okay, so it explains about 56% or 57% of the variation in the data. Okay, so that's good. So we have built the model. Now let's do the testing on this model. So what we are doing here, okay, first part is we are extracting this summary LM mod, right. So this summary will give you the full summary here, and we are extracting this explained variation. Okay, so this number, as I said, this 0.5657 is we are extracting in summary from as this command, right, summary LM mod 91, right. So these are some of the structures that is something you can explore a bit, right. So here is the adjusted R square. So you can see all of these carefully, right. So 1 is this, 3 is these features that we have, 4 is the estimates coefficients, 5 is the yeah. So all you have all of these values for degrees of freedom, R square, just R square, and then we want the adjusted R square, right. So this is something we want, and that's what we are storing in the EXPV, that is the explained variance in the data. How much of the variance in the training data that this model explains, right, adjusted R square, which is 0.5657, right. So 56 to 57% of the variation in the data is explained by this model. Now what we want to do is, now we want to predict on the test data, right. We want to use this model and we want to predict, right, how well this model performs on the test data. For doing that, the command is simply predict, take the test data, and what you should do with NA data, right. If you have NA values, what should you do? Just pass them, right, do not use them. So that is the command predict, and store these predicted values in this preds variable. Okay. 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 So it has like, okay, there is some sort of warning, but it should be okay, and what you will get is the predicted variables. Okay. So for some you have NA values, of course, that is something we cannot avoid. We can look into other issues, right. Maybe there are some independent variables that are a lot of NAs, etc. So that is something we will probably deal with later on. Okay, so what are the actual values? These are the actual Y values, right. This is something we have seen, right. These are the actual values, actual DM YPD values that we want to do, right. So what we can do, let us say we can very simply, okay, and also see, right, how well this matches. Okay, so of course these predictions are not very good, right. Okay, so maybe we can scale them. Okay, as you can see, the predictions are not that great, which we will see in a moment, right, when you calculate the R square, right, how much of the variation is in the predicted, in the trace data set is being explained by the model. Okay, so as you can see, there is almost, I mean, the correlation is likely to be very, very poor between the actual model and the actual data and the predicted data. Okay, so we can also look into the correlation values, right. We can say correlation okay, its p value is 0.98 and the correlation is 0.002. So almost no correlation, right. So that is not a good thing. Okay, which means the model is not actually doing very good prediction. And what could be the reason for that? We will talk about that in a bit later in the subsequent classes. Okay, and what could be the reason? Okay, for now let us focus on this and let us calculate this R square on the test data, right, that R square that we just talked about. For doing that, right, what we are doing is we are creating this data frame, right, C bind, this column bind, actual values and predicted values, and then we are taking only those actual and predicted where we have non NA values, right. If you have NA, then we are going to take those. Okay. And then what we are doing is we are simply calculating this R square value. Okay. So, this R square value is, we use this formula here to calculate this R square value. Okay. So, R square value, right, as you can see, you can run this carefully. So, as you probably understand, we are omitting NA, right, and then taking this Pareto minus actual, their square value, right, and then dividing by the length. Okay. So, how many observations we have, and similarly, right, we are taking this actual minus mean actual, right, and we are calculating this R square. So, if you look into this R square calculation, right, this is the formula, and this is what we are getting. Okay. And the R square is minus 3.28. Okay. So, this is not a very good model anyway, right, and this is something we already knew right here, because as you could see, right, the correlation is actually very, very poor, right. The correlation is 0.002 here also in the plot. As you can see, the correlation is, we cannot see we cannot see any correlation here, and the R square value is very, very poor, is minus 3.28. Okay. So, what is happening now, right? This is something you might be wondering, right. So, we have explained variance, right, we have 0.565 is like 56% of the variance is being explained on the training data, but on the predicted model is only minus 3.28. Okay. So, minus 3.28 means it's not explaining any of this variation, right, it is a negative value, and this is something that's a big concern for us. And you might ask, like, how this could happen, right. On the training data is doing so well, and in the trace data it's so bad. Okay. And the reason is very simple, is that when you take the training data, it's actually, because there is a huge number of features, what it's doing is taking all the features and trying to predict the values like very, very accurately, right, the y values or the dependent variable values. And for the training data, it can optimize these parameters, these coefficients and the bias, etc., all these terms in such a way that it can predict the y values very, very well. On the test data, it did not have that opportunity, right. So, it did not see the test data before, and it did not have that opportunity, and this is something that resulted in this very poor predictive model, where R square is negative. What it means is that in this training part, we have overfitting. Okay. The model is using a lot of these features, and it's actually doing overfitting. It's trying to predict or feed the data very, very well in the training data, so well that we cannot generalize across different data sets. Okay. And this is something that's not very informative. So, this problem of overfitting is there in many predictive modeling, and we have to address this. Okay. Now, you might ask, what could be the reason of this overfitting here? The reason is very simple, is we have a lot of features, independent variables, that are very that have very strong correlation among each other. Okay. And as I mentioned, this is the problem of multicollinearity. We can explore this a little bit, right. Some of the features that we have in the data, full data, you will see they are likely to be correlated. Okay. And there could be very strong correlation. Okay. So, I will take one or two features, and we will see like how these features actually are likely to be correlated. Okay. So, let us look at this feature, number of regulatory TF, right, how many transcription factors bind to the promoter region of a gene. This is simply a number, and then we can also look at, for example, this number of TF sites or transcription factor binding sites. Okay. There could be something as like number of overlaps in binding sites or number of sites. So, there are a lot of features that are actually correlated. Okay. Very strong correlation among each other, and this is something what we call as multicollinearity. So, let us look into some of these features, right, and see how well they are correlated. Okay. So, Okay, so let us look into the correlation between these two features. Number of regulatory TF is extract. So, this is from a database, and then we have the number of sites, right, number of transcription factor binding sites in the promoter region, and here the first one is the number of regulatory TFs that are known to bind a promoter of a gene. Okay. And you see there is a very strong correlation, right, 0.76. This is something we have learned, right, how to calculate this correlation coefficients. Here is the correlation 0.76 and p value is very significant p value, right, very strong correlation, strong positive correlation that we have between these two parameters. Okay. And this is not just one case that we have. We have, let us say, another variable that we see also that is very strong correlation, right. So, there what it means is that we have a lot of features that have very strong correlation among each other, right. So, these independent variables, they are likely to be very strongly correlated with each other. This is the problem of multicollinearity. What it does is that it actually increases, right, the chance of the overfitting, as well as the estimation of each of these coefficients is going to be not very robust, which results in this kind of situation where we are able to predict, or where we are able to explain the variance in the training data, but we are not able to predict the values in the test data very well. Okay. So, this is something which we will be talking about in the next class. So, I have explained the problem of multicollinearity, and this is something that we will be addressing in the next class. Okay, so let us now go back to the presentation, right, that we have talked about. So, we have looked into the generation of training data set, right, how we can generate training data and test data. I have shown you just one way of doing it. There are other ways of doing it as well, right, again using this DPL wire package, right, how we can separate out the test data and the training data. We will see that maybe in a subsequent class. We have learned about the predictive modeling, right. So, what we do is we take the training data and we build the model, right. We use the same LM function here, but not on the full data, only on the training data. What it does is it tries to build a model, right, try to estimate those coefficients and the bias for each of these features, and then try to best fit the y values that we have, or the dependent variable that we have. In the process, we get something called adjusted R square that actually tells us about the amount of variance that is explained by this model. After we have built this model on the training data, what we do is we apply that on the trace test data set, and we see how well this model predicts the values of the dependent variable on the test data set. And finally, we calculate, we look into the correlation, and we also looked into something called the R square, right. That is the predicted R square. It is like how well the model performs on the test data set. And as it should be as close to 1, right, to 1. So, if it is value close to 1, that means the model performs extremely well on the test data. So, this is something that has not happened, right. We have seen our R square on the test data set is negative, right, which means this model is not able to predict anything on the test data set, right. So, it is a very bad predictive model. What is the reason, right? What is the reason for that? We explained, right, what could be the reason, and we have seen some of these correlations in there. Is that many of these independent variables, they are actually correlated with each other, and that leads to something called multicollinearity, which can lead to problem of estimating the coefficients correctly, and we also are likely to have the problem of overfitting, right, where we are predicting, or where we are fitting the training data very well, but we are not able to predict on a test data set, right. So, this model is unlikely to be very helpful for any predictive purpose or identifying the key features, right. So, as I mentioned, that one of the major goals of doing this hands-on and doing this predictive modeling is to identify the features, right, that are likely to be the key drivers of these processes, right. So, some of the biological process that we want to understand, we want to identify the features that actually predict this process. So, here in this case we want to predict, for example, gene expression noise, and we want to see which feature actually express or it can explain or predicts the amount of noise a gene will have. Okay. And if your model is not very good, you cannot do that, right. So, we cannot identify these key features here without a very good predictive model. So, this is something that you need to address, and we will see like if we can improve this model so that we can build a good predictive model and we can identify the key features from the model, which will tell us something interesting about the biology of the system, right. This is something we will see with this example, but of course, you can imagine, right, very similar data sets we can learn a lot of new things. So, to summarize, what we have seen is we have so in this class we have introduced the predictive modeling part, and what I have shown you is that we can use linear models that can be used for prediction. We have seen that we can build this training data set, test data set, and then we can run these models for prediction. Of course, here the performance was not good, but we will look into the steps how we can improve this performance of these models. Another point I should mention is that this modeling that we did, right, with training data and test data, we have just done it once, but this is something that we also do repeatedly, so that we can calculate these adjusted R squares and then how much these values vary with different training data sets and test data sets, right. So, this is something that is very important. If we build a good model, we can identify these key drivers of a biological process. This is something that is the goal of these predictive models, and as I mentioned, right, these models are the performance of these models is clouded by multicollinearity, and we have problem of overfitting. So, we will talk about this multicollinearity in the next class, and we will see how to address this in an effective way. Thank you.